Hello there, my name is Robbie Wilson, and I'm a postdoctoral researcher in the Department of Chemistry at MIT. In this short presentation, I would like to outline a novel framework for the capture of carbon dioxide directly from air by exploiting the CO2 fixation machinery from photosynthesis and its implementation in synthetic biology platforms. In a year featuring one catastrophe after another, we could be forgiven for forgetting one of the earlier disasters that occupied the news cycle in late 2019 and early 2020, when huge areas of Australia were burning out of control. These bushfires were the largest in decades and widely recognized to have been driven by record-breaking heat and drought, an event which is growing in frequency and severity with time. During the most recent season, fires across much of eastern Australia saw over 46 million acres of land go up in smoke, creating a cloud of haze visible from space. These fires released over 800 million tons of CO2 into the atmosphere, a staggering figure. However, in an amount comparable to the total CO2 emissions from human activity in Australia from the previous year. Regrowth in burned areas are expected to mostly recover the emissions from those fires, whereas emissions from fossil fuels contribute to a persistent increase in global atmospheric CO2 levels, with 33 gigatons of energy released, related CO2 emitted in 2019. Humans pump out more than 40 of these bushfire events every year, year after year. We burn immense amounts of fossil fuels to power civilization, and this consumption is unlikely to decrease over the coming decades. Action to reduce CO2 emissions and straightforward methods to improve CO2 sinks globally, such as reforestation and improved land use efficiency, are vital components in our struggle against climate change. In addition, it is essential to counter anthropogenic carbon emissions through revolutionary technology aimed at permanently fixing CO2 directly from the atmosphere. Realistically, this will require billions of tons of CO2 capture in the form of ongoing and massively scalable carbon-negative technologies. Current engineering initiatives for direct air capture, or DAC, have mainly focused on chemical sorption technologies, where air is forced over a reactive substance that forms a covalent interaction with CO2. Subsequent heating of this material can release near pure CO2 that can be stored and fixed permanently by injection into rock, or perhaps more cost-effectively by conversion into renewable fuels. Several major limitations to this technology include cost-effectiveness, with recent estimates of $100 to $600 per ton of CO2 fixed, the scalability of such large, sprawling industrial capture sites, and the carbon cost behind materials and site construction, energy supply, and carbon product utilization, which may prevent its use as a carbon-negative system. By relying on chemical sorption strategies, the failure of this technology, or scalability, or economic and environmental unfeasibility would be catastrophic for climate remediation. Investiture in novel and radical carbon capture platforms is essential to hedge against these failures and maximize our ability to achieve a workable and realistic technology to counter CO2 emissions. In nature, the vast majority of inorganic atmospheric carbon is fixed into organic carbon through the action of photosynthesis. In this process, light reactions convert energy from sunlight into chemical energy, which is then used to power the Calvin cycle, or dark reactions of photosynthesis. A key enzyme of the dark reactions, and the enzyme responsible for CO2 capture, is ribulose 1,5-bisphosphate carboxylase/oxygenase, or Rubisco for short. This carbon is then sequestered into the biosphere as sugar, which is then converted into all forms of carbon found in biological organisms. In fact, all of the carbon present in oil and coal was originally fixed millions of years ago through Rubisco. Here I propose that systems utilizing Rubisco will be essential again in the recapture and permanent fixture of atmospheric CO2. Removal of CO2 from the atmosphere is a challenge for any technology, man-made or natural, due to its low abundance, currently 415 part per million, which is generally seen CO2 sequestration technologies implemented at point of source, such as in the flues of power plants or in ammonia production. The catalytic mechanism of Rubisco must also balance this relatively low abundance of carbon substrate against the far higher concentration of oxygen at some 200,000 part per million, which the enzyme can also react with, albeit unproductively. In each scenario, the Rubisco enzyme binds to a 5-carbon sugar substrate, which is reacted with CO2 to generate 2 molecules of glycerate, or a molecule of glycolate if oxygenation occurs. The product, 3-phosphoglycerate, is a component of central metabolism across life and can be incorporated into these pathways when Rubisco is expressed in alternative hosts. Rubisco has been used in these alternative hosts and non-photosynthetic organisms to capture environmental CO2 for several decades. These platforms have been used to engineer improvements to Rubisco carboxylation using techniques like directed evolution by the common laboratory bacterium Escherichia coli, which is often used as a surrogate due to its fast growth and ease of use. In these systems, mutant libraries are screened for improvements in Rubisco activity, which can rescue the bacteria from a growth deficit, thus correlating CO2 fixation activity with improved bacterial growth rates. More specifically for the context of this talk, Rubisco have been expressed in various industrial yeast strains, which has permitted greater yields of carbon products such as ethanol due to the recycling of waste CO2. These microbial platforms generally only require the introduction of Rubisco genes and the enzyme PRK, which produces Rubisco substrate. An exciting development of this technology was outlined in 2 recent papers where E. coli and the industrial yeast Picia pastoris were engineered to permit growth on CO2 as a sole carbon source for biomass growth. The ability to use CO2 in this way was achieved through the expression of Rubisco from enzymes from bacteria and the use of formate and methanol to power these synthetic Calvin cycles without contributing any carbon to biomass. These landmark studies provide a proof of principle foundation for the large-scale usage of CO2 using synthetic biology platforms. However, due to the type of Rubisco used in these platforms, efficient capture of CO2 from air is impossible, and thus they require high CO2 concentrations to survive. Similar Rubisco types, when used to replace the endogenous Rubisco of chloroplasts, likewise require a high CO2 environment for growth and cannot survive in air. Many different types of Rubisco exist, and they show diversity in their form and function, which are adapted to their host environment. Rubisco from plants have evolved to capture CO2 from air fairly independently, and their enzymes show a kinetic affinity for CO2 that is unrivaled by most other forms, such as a low Km for CO2. While at the same time, they're also fairly insensitive to the presence of oxygen, i.e. having a high specificity value for CO2 over O2. These parameters appear to be in a trade-off with the speed at which the reaction with CO2 can occur. And so plant Rubisco tend to operate within a narrow range of a few carboxylation reactions per second. To offset this trade-off, many other organisms have evolved specialized and complex CO2 concentrating mechanisms to increase CO2 availability around Rubisco while preventing reaction with oxygen. Examples of these include the liquid-liquid phase separating pyranide component of green algae, or the carboxysome of cyanobacteria, a protein fortress encapsulating the Rubisco that lets CO2 in but keeps oxygen out. In line with these environments, these enzymes have evolved to be much faster than plant forms, which show greater sensitivity to oxygen and need high CO2 environments to efficiently capture CO2. Similarly, bacterial Rubisco are associated from organisms that inhabit environments naturally high in CO2 or devoid of oxygen, such as in the deep ocean and other extreme environments. In consideration of these origins, plant Rubisco are therefore the best in class for deployment in microbial hosts for the purposes of direct air CO2 capture, without the need for additional systems, compartments, and unknown factors. Rubisco from eukaryotes, like plants, are not simple to express in alternative hosts. For many decades, the highly efficient enzymes from plant leaves could not be recombinantly produced in organisms outside of the plant host. Recently, my previous work, in collaboration with colleagues in Germany, overcame this limitation by co-expressing a number of molecular chaperones alongside the Rubisco genes in the common bacterial strain of E. coli. These chaperones form an assembly line of helper proteins that allow translated Rubisco to fold and assemble into the correct three-dimensional shape so it can perform the carboxylation reaction. Production of these components is essential to permit high levels of plant Rubisco expression in an alternative host. High levels of production can now be achieved in E. coli, as shown by this gel of protein content of bacteria expressing all of these factors, where the blue coloration represents the concentration of Rubisco protein amongst all other proteins in the bacteria separated by size, and with lane 2 showing no Rubisco expression and lane 4 showing a high production strain. The protein from these cells also show the same kinetic properties as leaf synthesized Rubisco, and it is able to capture CO2 at high rates under optimal conditions. To date, the efficiency of carbon capture of plant Rubisco, when expressed in these kinds of hosts, has not been measured in vivo, but incredible progress in the field is likely to put a number on this soon. Integrating this expression within the wider context of synthetic biology may produce a truly transformative platform for CO2 remediation. My vision for this technology is for biological CO2 assimilation machinery to be partnered with metabolic pathways that allow for the conversion of 3-phosphoglycerate to sustainable carbon-based end products. Historically, the most obvious target, and one for which the metabolic pathways are already well characterized, would be biofuels. Although this would allow for the production of low-carbon fuels, unless crude oil prices are high, biofuel production will never be competitive. Instead, pathways for the production of raw materials that can act as carbon-rich replacements for things like concrete, steel, and wood in construction are far more viable long-term sinks for atmospheric CO2. Ultimately, atmospheric carbon must be permanently sequestered in the earth from whence it came. And in this case, these non-biodegradable materials, exhausted of their utility, will be buried in landfills and replaced. Overall, the tools from photosynthesis represent a first step in a multi-stage endeavor towards a truly carbon-negative platform. The second phase must evaluate the best conversion pathways for useful end materials. Innovation of carbon-based materials that can utilize CO2 conversion in the most optimal and safe fashion must occur in parallel with to assure the timely success of these platforms. Inevitably, we will need to provide an energy source to power carbon capture by Rubisco, analogous to the light harvesting reactions of photosynthesis. Due to the flexibility of synthetic biology, there are a multitude of other pathways from chemotrophic organisms that can be harnessed to obtain energy from the environment. This flexibility may allow synthetic hosts to utilize a variety of cheap energy sources to power carbon capture. These include sulfur and ion oxidizing bacteria, and possibly the grand ambition of electrosynthesis, where electrons are utilized directly. I believe that these efforts to remove atmospheric CO2 need to be commercially incentivized, and considering the sheer volume of CO2 that must be removed from the atmosphere, commercial viability should be a guiding principle through which we develop these platforms. So what kind of performance might be expected or required for such systems to capture atmospheric CO2 at scale? Even under optimal conditions where energy and CO2 were not limiting and ignoring effects of respiration, synthetic biology platforms would, under rough estimates, need to be cultured at scales of hundreds of billions of liters in order to capture a gigaton of CO2 each year. That's in the order of 150 to 200,000 Olympic-sized swimming pools. Although a huge figure, as a frame of reference, each year we produce approximately 200 billion liters of beer, which shows that the industrial capacity for these endeavors is at least feasible. So you may be asking, if we're going to go to all this trouble of taking CO2 capturing machines from photosynthesis, and you need a clean energy source to power CO2 capture, then why not simply use existing photosynthetic microbes to do the job? Well, some people are, such as synthetic genomics in California, and these are vital possibilities to explore while we search for the best scalable technology to get the job done. However, there are some advantages to a purely synthetic biology strategy. Firstly, algae require abundant access to light that results in large surface area requirements for efficient carbon capture. Probably the most important factor, however, is the ease of genetic manipulation and efficiency of DNA transformation of lab E. coli and yeast compared to microalgae. And these have metabolic pathways and responses that are extremely well characterized. This is particularly important when considering the extent of genetic engineering required to convert CO2 into useful materials. Common industrial strains are also supported by an entire bioreactor technology that has been developed over decades specifically for these organisms, and which may not be compatible with organisms such as diatoms, cyanobacteria, and other green algae. Finally, photosynthetic organisms contain an entire history of evolution surrounding CO2 utilization programmed into their biochemistry. Often these organisms will negatively regulate enzymes involved in CO2 sequestration under conditions of elevated CO2 or low light. These regulatory networks may be significant barriers to obtaining efficient conversion of CO2 into useful materials. In closing, most of the current carbon capture and storage technology is viewed through the lens of industrial chemical processes, which is dominated by chemistry and engineering researchers, who will inevitably provide chemical and engineering solutions to the problem of CO2 capture. Biology is an integral component of the carbon cycle on Earth and provides numerous tools from photosynthesis that can be leveraged to enable the CO2 capture at scales competitive with current research streams. In addition, synthetic biology allows for the enormous variety of chemical transformations that occur in biological organisms to enhance CO2 capture and, more importantly, convert that carbon into value-added material resources, which, In the dream scenario, would provide a commercial incentive for scalable and profitable CO2 capture and conversion. Overall, the challenge is immense, but so is the problem we face with continued CO2 emissions, and the stakes could not be higher. Increased frequency of extreme weather events, rising sea levels, drought, crop and agricultural insecurity. We're sleepwalking on a slow march towards a bleak future. To what extent is our current experience of climate lagging behind current CO2 levels? We cannot afford to wait and see. We must act now in the development of new technology if we hope to have a carbon negative future. So I'd like to acknowledge my host lab for supporting my work on Rubisco at MIT. I'm currently situated in the Department of Chemistry in the laboratory of Professor Matthew Shoulders. Thank you for listening to this presentation, which I know might be quite different to the style of science attendees in this conference are usually exposed to. I've tried to frame this for a general audience, but if there's anything I can do to clarify or questions I can answer, I'm happy to do so during question time, or you can leave a comment on this video and I'll do my best to answer them. Also, if you have any ideas which may be helpful, such as carbon products or materials that might be attractive to synthesize using synthetic biology, then I would love to hear about them in the comment section. Cheers, and thanks once again.